Okay, thank you very much, Anat. And definitely a very excellent project that can enhance the collaboration between the hospitals and the university at the students level. And we're moving to the next talk by Professor Gozes. I think it's this one. Is that yours, Illana? Yeah.
So I'm not sure if it was Bugs Bunny or one of the other of Walt Disney characters that said, and now to a totally different subject. So I will tell you about my own travel going from the bench to clinical development. So and actually everything can be taught in a course because I took several courses on the way as I was doing it. But I will start with some science. So this is me, and this is.
Can I say who said it, by the way?
Excuse me?
Can I say who said it?
Yes, please.
It was Monty Python.
No.
They say now for something completely different.
But I think it was copied by Walt Disney in one of his movies. I have a feeling. Okay. So the idea, if you want to go like from your medical or basic research studies to be a biotech, to go to the biotech industry, you first have to have an idea. And so this is translational research. You have to have an idea, and then you have to decide if you want, and this is now in biology, if you want to go to the diagnostic field, maybe then do some very innovative MRI studies, or if you have a molecule, sorry, that you would like to develop. So my idea was that I would like to develop some molecules because I was actually trained as a biochemist originally. So I looked at the brain neuron-glia interaction, and we isolated new proteins then to science. And then I had, you don't hear me. And then I had another idea that maybe if these new proteins that are providing nerve cell protection, so I was looking for nerve cell protection to protect like Alzheimer's disease. And the other idea was that if I can cut them into small molecules, maybe then I'll have small molecules that one can actually give to a human being. So that was the original idea many years ago. And then in neuroprotection, if you want to protect the nerve cell, the major protein in the nerve cell is called tubulin, and it makes the tubes, the microtubules. And so I thought that this is a good target to protect. And fortunately, the compounds that I was developing actually interact with the microtubules. So I have compounds that provide protection and interact with the microtubules, and through the microtubule provide protection. So this is still basic science. But I have to go, actually I should go back. Sorry. So you have to have a compound and you have to have a target. You can actually start with the target. There are many companies that are based around the target, and then you go to the compound. And then I knocked out the protein itself. I got a model that actually doesn't learn. So you have to have, in order to go from the basic idea, from a molecule that you tested in tissue culture, you have to go to the proof of concept that you had on your slide to animal models. And the more animal models you are using, the better off you are. Because right now, we are all working with transgenic animals, and in truth, these are inbred strains, so we are working with one animal. They are different because each one is in a different part of the cage, but we are actually not, it's not mimicking exactly the ideal situation, the human, that we are also different. So we have to go from the cell, from the molecule, from the biochemistry, to the cell, and then to the animal, and this is called this is actually the pharmacology side of drug development, and this also needs to be in a way learned. And so here, this is this is a test for memory in a water maze, and you see when the animal learns, when the animal doesn't learn, it doesn't find the platform. When it does learn, it does find the platform. And the pathology is associated actually with the pathology of the microtubules. I won't go into it because it's late. But the major point that I want to drive at here is you have to find a target. you have to have a molecule that identifies the targets and protects against the deterioration that is associated with its target. And here I'm showing you this is at the level of the cell, this is in the level of the entire animal, and you can do now with the advantage of MRI, which wasn't there when I was a student, nor was molecular biology. You can actually go and you can measure movement of molecules inside the brain. So you can measure axonal transport now, you can measure the brain structure, and you can really develop, and also the microscopy is much more advanced than it used to be, so you can really develop molecules and understand exactly the molecular mechanism by which they are working. So, and this is important because when you continue and you want to do the translation, you actually have to justify why do you want to do this translation, why do you want to go into humans, and then you have to show specifically what is where did it work. As I said, many more models is better, and how does it work, and how is it different from other compounds that every one and his neighbor are doing. So this is like showing different models, and here it's protection against tauopathy, which is one of the major pathologies in Alzheimer's disease. And this is just to show that the protein that we discovered is not only associated with cognitive decline, but also with autism. So this is by doing the genetics, but it's a bit irrelevant. So the other thing, that once you have the molecule, or maybe a family of molecules, or you have a target and you want to develop the molecules against it, you have to go through very specific stages of drug development. And the specific stages of drug development is, firstly, if you have a molecules and you want to treat the brain, you need to know that it gets into the brain. So you have to develop technologies that show that the new molecules is inside the brain. So basic scientists usually label the molecule, and then they will look in the brain and they see the labeled molecule. But the labeled molecule is not exactly identical to the non-labeled molecule because you changed it, because the label is changing the molecular weight and the properties. So you actually have to measure the molecule itself. And now with the advantage of high-pressure liquid chromatography and mass spectroscopy, you can actually measure small molecules, and you can measure them in the brain material, you can measure it in blood, you can measure it in the CSF, and you can actually do the pharmacokinetics. And this is done usually not by the basic scientists, but by companies that this is what they do for living. And another thing is, after you have the proof of concept in your laboratory, it actually has to be verified maybe in other laboratories to make sure that what you are talking about is right. And then this is like a cookbook. You have to do toxicology. So it's not enough that the drug is helpful. You have to know, מירה, יש לך שאלות? טוב. אז you have to also know exactly that it doesn't do any harm. So the Food and Drug Administration, for example, in the United States, will ask you what are the side effects. So this is very, very important to know exactly what to do. And this is like a cookbook, and one has to go exactly with rules and regulation, and actually test it in many, many animals before it can in many animals just for toxic side effects, before it could go into the human. And this is a whole occupation, actually. So it's not enough, and I don't know how much physicians are aware of all the animals that are killed on the way to the clinic for a new compound. Another possibility is to actually take a compound that has failed in something else, but is safe, and see if it will work in other indication. So the pharmacology, the pharmacokinetics, how does it get into the plasma, into the CSF, and into the brain is very important. And then you have to have a set of toxicology studies that have to be done, and this can be taught to the students who want to take this route of career. And then once you have all this prepared, you have the pharmacology, the preclinical pharmacology that you know that it's working, and it's going to be the drug for Alzheimer's disease, and it does not have any side effects, and it does reach the brain in amounts that are enough to actually be active, then you have to try and think how are we going to develop it. So then you have to write some kind of like an executive summary of what do you have and where do you want to go. And this is, after that you have to write a business plan. Or you go to the university authorities and you say that you would like to maybe license it to another company that knows how to develop it. And what is also very important is that if you don't patent your invention, because this is a new invention, then you won't be able actually to develop it, because this is where you move from being a scientist to become a business person, and you cannot sell it. Because if you develop something that is already known, then the company that is developing it is not going to get the money back for the drug development, which is very expensive. So actually, intellectual property equals patents, and you need to patent everything on the way to development. And this is what is being commercialized. So this is the point where you move, once you start the clinical development, you actually move from the academic, complete academic world, and you move a little bit into the business side of it. So you need to have a lot of publications in the best journals in order to push it forward. And then you go and you have to plan the clinical trial. Now, this is something that many people can fail in, because you have to choose the right indication. You wanted Alzheimer's disease, let's say, but most of the clinical trials in Alzheimer's disease actually fail, and maybe they fail because it's too late, and if you want to do neuroprotection and all the nerve cells are already dead, you can't resuscitate them. So you have to be very, very careful in thinking together with the physician, and that's where the basic science meets the physician, and really give it a very hard thought to where to take this very, very promising compound that worked so well in so many animal models. And then you have to think about the patient population. Writing a clinical protocol, as my physician friends know, it takes a long time, and it has to be very thoughtful. You need to know who you are going to include, who you are going to exclude, and then actually all the clinical trials that are going on are written in a site of the FDA that's called clinicaltrial.gov, and you can actually see. So if somebody is interested, then you can check what is going on in order to know that you're not going to repeat something that your neighbor did. So what happened to the compound that we took forward? So I did these steps. I actually went to a course in Europe to learn how to write a business plan. Not that I know how to do it, but I went to learn. And the major thing is the executive summary. And so you see, so the proof of concept, safety, and pharmacokinetic. The proof of concept are those experiments in animals. And then the safety are all these toxicology studies. And then the pharmacokinetic studies. And then you actually have to give the drug for 60 days, or for as long as, 60 days is already close to enough, but you have to go also higher with the dose until you see a dose that is toxic. And if you don't see, you continue going higher until you cannot dissolve your compound anymore. You have to see, if you want to treat the brain, you have to see that it penetrates the cerebrospinal fluid. And you have to go for healthy. If you want to go on Alzheimer's disease, you still have to go to the young population to make sure that nothing happens. Then you go to the aged population, and then you can actually reach the clinic. And so in our case, there were 35 proof of concept studies in animals, and many were not done in my laboratory to make sure that it's reproducible in different laboratory. And with the compound, it was in already 710 subjects, and I'll show you the good results. I'll tell you also something about the bad results. So I'll start with the bad news. So there was a phase 2-3. So the phases of clinical studies, there are several phases. So the first phase is just to measure if it is toxic in people. So that's done in healthy volunteers. They can be young. Even if you are going to targeting Alzheimer's disease, you have to see that it doesn't do anything. So that's a very short acute application, and you measure that the compound and actually this is a profession. There are clinical trial monitors, and they learn, there are courses to actually become a clinical trial monitor, and this is what they do for a living. So somebody that finished brain studies and wants to become like a BSc and wants to become a clinical trial monitor, that's possible. And I know actually people with a PhD that became afterwards clinical monitors. So in terms of the clinical development, you do the phase 1 in young people, then you go and you do it again in volunteers, in older volunteers. You also do the pharmacokinetics in volunteers to make sure that the drug gets to the blood and maybe gets also to the CSF. And then so the trial that didn't work was already in phase 2-3. So phase 2 is the proof of concept for activity. So after you know that the drug is totally safe, doesn't do any harm, or at least not a harm that you have to write home about, then you go to the second phase to look to see for activity, for biological activity, for what you've seen in the animals, to see if you see it also in people. And we went to people with mild cognitive impairment before Alzheimer's disease, and we also went to schizophrenia patients, because in both situations you have problems with the microtubular system, with the microtubules. And we thought that this is what we would like to repair. We also went to a disease that is terrible, that's called progressive supranuclear palsy, that developed very, very rapidly. And I think that there it didn't work. And there I think that we went there a bit too late and maybe with too high dose. But what we did get out of it is a paper in Lancet Neurology, which is a nice journal. So in terms of phase 2, so phase 2 is proof of concept for activity in people. So you have to take a large enough, you have to have a statistician go with you all the time. And if you want, you can, of course, study statistics. I need to finish. Okay, so I'll finish. I won't explain. So here we got statistically significant results by after 3 months of application. You see the people remember better after they got the drug, and in two tests. And here we got also statistically significant results in a cohort of schizophrenia patients. So choosing your patient population and giving the right drug, and the right dose is very, very important. And this is something that one has to learn. And then you have to have a pipeline in order to continue. So this is it. I was told to finish very quickly. So we want to save this brain and make it like that. So, and we have to be educated both at the science. You have to know the science, you have to know the business, and you have to know statistics, so not only brain, and you have to be actually a physician in order to develop it. And also talk with the FDA, that's in the United States. In Europe, it's actually the EMA, but many trials are actually done globally, so you have to understand all the authorities everywhere. Okay, so...
Taking into account your trip and experience, and now bringing it back to Tao's students, what would you do different to make your life easier next time?
So I think maybe it all boils down to people that this I'm telling it straight out of my sleeve, but I think you have to really, every step that you do, you have to think very carefully, and I think the patient population is extremely important. I have to say that the journey was excellent. I mean, I really enjoyed every minute of it, almost, except and it's very important to work always with the right people. And I think that maybe also make sure when you are successful, so I have a poem that I really like, and it's by Rudyard Kipling, if you meet with triumph and disaster, treat those two imposters just the same. Because if you know, when we were successful with the MCI trial, the guy stayed and drank wine the whole night. And I mean, it's good, but you also have to be humble and make sure that you don't, you don't become too big-headed, and you have to be very, very careful how you go forward. And like with the progressive supranuclear palsy, it may have been a mistake, even though I thought originally that it is a genius. And why did we go after this indication? So there is where you have the grid and the cleverness, because if it is an indication where there is a very rapid change, then you can say that you have, for like brain diseases, that neurodegenerative diseases that develop very rapidly, then you can actually see disease modification. And I don't think we have too many drugs that are disease modifying at all in all these neurodegenerative diseases. So if you go to a disease that develops very fast, maybe you can see modification. But that's very difficult. Why is the disease developing very fast? We don't know. So that's a danger. And also going to a disease that is very rare, you get from the FDA and from the EMA, you get actually some perks. You get orphan indication, and which means that you have the whole market for yourself for this specific indication. So this is a wonderful idea, and then you will become very rich. But on the other hand, you have to be very, very careful. And we did this mistake, and Famos, a company in Israel, a long time ago, did the same mistake. And so one has to be careful, and one has to go more, a little bit slower, and look around and think, and the indication and the dose and the timing. But that's for the clinical development. So this is something that we can teach, and this is probably something that I would have done differently. The other thing is that I started the company in the United States because we thought that we'll get more funding there, and then it moved to Canada because we actually got more funding to Canada, but I ended up going 4 times a year to Vancouver, so that wasn't that easy. So maybe next time I would start the company here in Tel Aviv.
Okay.
No questions?
Thank you very much for sharing this fascinating journey.